welcome back to the course in Animal Physiology in NPTEL. So, we are into the section 4 and this will be the third and the final lecture of the section 4. So, in this section we are talking about the circulation process. So, we have already discussed about the different vessels, their cross sectional area and how the pressure changes, how it is flowing once the blood is being pumped from the heart. So, if you remember, when we drew the first overall outline of this section, we talked about the blood is flowing from the arteries to the arterioles, then through the capillaries to the venules and to the veins, okay, and back. And we talked about the cardiac output and okay, and the pressure differences. And in the section of the can, we talked about that we will be discussing the hormonal and neural control of this whole dynamics at some other point, while we will be talking about once we are finished with the neural and the hormonal part of it. So, today we are going to deal with this part, the capillary and the capillary exchange. So, before I move on to the capillary and capillary exchange, while talking about all the different pressures what we have talked about, I just wish to ensure that you people are aware of sphygmomanometer. This is the one which is used by the doctors to measure the pressures and the pulse. Just to update you on that once if some of you are not aware of it, it is a simple device. You have seen the doctor kind of put it there and kind of check your pulse and everything. So, this is the device which is being used. Okay, it has a valve which can see the see the undulation of the blood vessels, and it can measure and it can hear to the noises and can give a whole bunch of informations. And you talked about that lup, dup, what we have talked about, all the 4 sounds and everything is being done with this sphygmomanometer. Okay. So, along with a stethoscope attached to it. Okay. So, now we will move on to the capillary exchange. So, let me give you before I get into any technical detail, let us see a practical situation. So, that will help you to appreciate this problem much better. So, whenever you have, say for example, honeybee bites you or some kind of wasp bites you, you see there is a swelling at some point or other. Wherever it bites, you see a swelling. Why there is a swelling? Did you ever ask this question? And invariably people or the doctors or some learned person, so there is an inflammation. So, that is why there is a swelling. What really is that swelling? And if you go to the doctor, doctor will use a technical terminology called edema. It is a edema likewise. And if you go through the dictionary, it says accumulation of fluid is called edema. So, why that edema occurs or why that accumulation of fluid occurs whenever there is some kind of insect bite or wasp bite or honeybee bite or XYZ situation, and there could be many other pathological situation, physiological disturbances during which it happens. But overall, in a normal condition, like I am standing here, I do not have any edema or any kind of inflammation at any point or any kind of fluid accumulation at any point. So, then what is this fluid accumulation? Why there is a fluid accumulation? Because if you realize while talking about, so this is something people have to realize first before I get into the technical details of these definitions, formulae and all those things. Those comes later. What is the first thing you have to realize, that there is a philosophical understanding of the problem, and that is very, very critical for you people to appreciate. So, if you look at the blood vessels and if you look at the way our network of blood transport is being designed. So, what is happening? From the heart, the blood is pumped out through the vessels, it moves along the arteries, and at the capillaries there is an exchange phenomena taking place. So, all the oxygenated blood is downloading the oxygen to the different tissues, and from those tissues or cells it upload the carbon dioxide into the vessels, and from there it comes back through the venules and the veins and the vena cava back to the heart. So, it is kind of a closed loop system which is continuously flushing every component. So, for example, this is me. So, from here, like the blood is moving all over the hands, all over the legs, all over the body, across the brain and everywhere, and it is continuously flushing it out. So, there is no room that any so whenever this flushing will stop, it is just like in your say for example, in your community or in your colony or wherever you are staying, there is continuously there is a sewerage or sewerage line which is continuously flushing out all the things which are being dumped from your house or XYZ houses. Okay. So, whenever there is an accumulation of fluid taking place, it means there is some kind of a leakage in some kind of a vessel, and this is very critical. There could be 2-3 possibilities. Either there could be a leakage, or there could be an abnormal situation by which the flow is cannot ensure that everything is being flushed out. This simple understanding is very essential to understand the capillary exchange process. So, as of now, if I go back, when I am repeatedly telling you that at the capillaries, like if this is a capillary network, imagine this is this is the capillary network where I was drawing that. If imagine that this thumb, one of this thumb is the artery, and this thumb, the other thumb on the other side is your vein. So, from the arteries it is coming, and in the capillary the exchange is taking place. So, what ensures that carbon dioxide will be taken up by these capillaries and oxygen will be given out by these capillaries? What ensures that some of the solutes which are present there will be given out and some of the toxins which are present there will be taken in? What are those forces, or what are those physical phenomena which dictates all that thing? So, in order to discuss this, I will take you back to my lectures in the membrane physiology section. There we talked about some of the forces. If you remember, we talked about osmosis, one of the forces. We talked about diffusion, the another forces. So, if you remember, in the diffusion I told you that basically from higher concentration to lower concentration. Say for example, you have lot of sugar out here and lesser sugar here in solution. So, from the higher sugar, the sugar will try to diffuse to the lower concentration of sugar. Okay. Then we talked about filtration. About filtration, we talked about there should be a pressure difference by which the filtration will take place. We talked about osmosis, and even in the last class also I told you the how to measure the osmotic pressure. So, if you remember, I showed you that U tube on a U tube. I told you that not the YouTube what we talked about in the net. This was the U-shaped tube. I showed you that from one side there is a semi-permeable membrane in between, and you are filling water on both sides, and water can pass through the semi-permeable membrane without any problem. But the condition is that the solute cannot pass through. So, if you have a higher concentration of solute on one side, one arm of the tube as compared to the other arm of the tube, so the solute concentration, the side or the arm where the solute concentration is higher, water molecules will try to move more towards that. And if you want to create a potential difference, if you want to balance the potential difference some way or other, then you have to give an equal and opposite force, which is in the form of hydrostatic force. So, these are some of the forces. So, let me enumerate: diffusion 1, filtration 2, osmosis 3. These are some of the forces which dictates, and we will come to the different values which will help you to appreciate why oxygen is being downloaded, carbon dioxide is being uploaded, and all these features. So, these are the simple basic forces which ensures that the exchange takes place. Now, coming back where I started this, I ask you, like, think of a situation when there is a fluid accumulation. During fluid accumulation, what happens is that the permeability, or because this is happening ensuring that the vessel permeability remains constant. Okay, you cannot change all the parameters. You can change one parameter and you can study the other parameters. So, because of XYZ conditions, the vessel permeability changes. It mostly happens, and I will again reiterate that at the end of the discussion when I will talk about all the forces. So, what happens whenever a toxin, whenever a wasp or a honeybee or an insect bites, it secretes certain toxins into your blood vessel, and that toxin from the arteries travel all the way to the capillaries, or from the veins it may go to the heart and comes back to the capillaries where the actual exchange taking place. And at the capillary, what it does, if you again recollect the structure of the capillaries, it has fenestrated capillaries, we have continuous capillaries and all. So, at the capillaries, what it does, it binds to the wall of capillaries and change the permeability or the exchange phenomena, and by virtue of which the body loses, or not the body, the vessel loses some of the more water which it cannot take up. And if I take you further back in my lectures in membrane physiology, if you remember, I was telling you this, there are toxins which can make channels. If you go back in the membrane physiology lecture, you will see I talked about some of the valinomycin, gramicidine, and in that section where I was showing you that there are circular peptides which can form channels through which the water molecules can pass in and out. And I talked to you about the gramicidine, and I specifically, I remember I drew the structure of valinomycin, gramicidine, and I talked to you that there may be a series of toxins, antibiotics, which has the ability to form a pore along any kind of a membrane, and that is exactly what a toxin does. When the toxin travels at the capillaries, where the wall is much more, I should say much more thinner, because it is almost a single red blood cell, it is around 8 micron. If you see the dimensions which I have already taught you people. So, at 8 micron diameters, basically it is almost one cell thick membrane like this. So, it creates holes along it. So, now what we will do, we will come back to this, you will be able to appreciate it much better once I tell all the forces which are involved and the generic calculation. Based on that, what I expect from by the end, in next 15-20 minutes, once I am done with all the forces, what I expect is that whenever you see a edema, whenever you see a accumulation of fluid, immediately these forces should come into your mind. What are the filtration forces? What are the osmotic forces? What are the diffusion forces? What are the reabsorption and everything? This should come straight without even a second thought. It should be your second nature. You should be able to immediately pick it up. So, it is basically these forces which are playing a role, and based on that you should be able to at least have some form of layman diagnosis into the situation. Okay. Now, let us enumerate the forces which are involved in the capillary exchange. Capillary exchange. Okay. So, the forces what we will be talking in this capillary exchange will be your diffusion, your filtration, second force, you have the reabsorption, and it is basically the interplay between filtration and reabsorption. This is the interplay of this which regulates the exchange of the fluid across the capillary membrane. So, just to give you some of the numbers, and so one more thing which I actually did not mention. So, I thought I better discuss that first. Okay. So, whenever I showed you, I have shown you this diagram. Okay. So, this is the one side is the capillaries, the arteries, arterioles, and on side is the venules, and here is the exchange taking place. At this exchange zone, along the blood vessel, there is a parallel vessel which runs. That vessel is called lymphatic vessels. We will come to these lymphatic vessels while we will be talking about the white blood cells and the immunity. So, how it looks like physically is, let me draw this, this will help you. So, if this is the artery side of it, and this is where, so this is the network of the capillaries. So, this is the capillary network. And the red is, okay, from here the venule network is picking up. Okay. Okay. So, the blood is coming like this, okay, and so these are the arterioles, and these are the venules. So, the blood is coming from the arteriole side, and here is the exchange taking place. So, let us represent the exchange by different arrows. Here is the exchange zone. So, at this zone there is a parallel vessel which is running, and I will represent the parallel vessel in black, so that you can distinguish it. Something like this. This vessel equally has a very, very fine mesh network. And this whole network is a parallel system along with the blood vessel, which is called the lymphatic system. This run parallel. So, whenever there is a fluid which is being given out, and this space which is in magenta, this space is called interstitial space. Interstitial space. And this black one is your lymphatic vessels. Okay. So, whenever there is a fluid which is coming out from the capillaries, it is being reabsorbed by the lymphatic vessels, and this is how the blood, it is being ensured by the body, the fluid does not get accumulated. So, as a matter of fact, almost about approximately about 3.6 liter of fluid flows out of capillaries to the lymphatics. To the lymphatic system, which is further this lymphatic system, what it does, it brings the blood back to the blood vessels. Okay? So, this is very, very important to understand that there is a continuous interplay of lymphatic system and the capillary system, interplay of lymphatic system and capillary system. And what it leads to, this interplay of lymphatic and capillary system ensures that there should be a continuous flushing action, which will continuously flush all the tissues. Okay. It assists in transport of very large molecules which otherwise cannot be really transported. This is the process which accelerates the rapid exchange of nutrient and moreover, it is the exact interplay between lymphatic, this is the balancing of the lymphatic and the capillary vessels, which ensures that we do not suffer from any kind of fluid accumulation or any other kind of situation. So, this is very, very important for us to appreciate. Okay. So, these are some of the major functions as a part of the interplay of lymphatic system and the capillary system. Okay. From here we move on to all the forces what we have already discussed. Now, we will come to the exact value. So, I am not again describing all the forces because I have already done that. So, now what I will do, I will redraw the vessel and I will highlight the different forces which are involved there. Okay. So, this is the blood vessel. Let me draw it in a way so that it makes okay. So, this is the venule side, a little bit more. Okay. So, this is the arterial side is taken and this is the venule side. Okay. Okay. Now, so here you have the arteriole. So, blood is coming from this direction, entry of the blood into the capillary network, and this is the venule. And imagine this zone is the capillary network zone. This zone is the both arrows I am showing is the capillary network zone. Now, there are 2-3 terminologies which I am going to come, and okay, let me. The first terminology is NFP, net filtration pressure. So, we will be measuring the net filtration pressure at 3 different zones: at the beginning of the capillaries, the middle of the capillaries, at the other end of the capillaries, where. So, if you look at the 3 zones, if you look at it, so this is the zone where capillary is involved in giving away oxygen. because this is oxygen rich blood which is coming, and this is the zone where capillaries are taking in carbon dioxide from the surrounding tissue. These are the two zones. So, how it does so? So, before we understand this, we have to realize the capillary hydrostatic pressure. So, I have already talked to you about this hydrostatic pressure. If you remember that, when you put an equal and opposite pressure in that, U tube kind of things, where I showed you that you put you give an equal and opposite pressure against the osmotic pressure, that is the hydrostatic pressure. So, what are the hydrostatic pressure? Capillary hydrostatic pressure, or the hydrostatic pressure of the capillaries is called capillary hydrostatic pressure. It varies from 35 millimeter mercury to 18 millimeter mercury in the capillary zone. So, at the beginning, so the capillary hydrostatic pressure at this zone is, so let us represent it by CHP. Capillary hydrostatic pressure at this zone is 35 millimeter mercury. Okay. Capillary hydrostatic pressure in between out here is approximately 25 millimeter mercury, CHP, capillary hydrostatic pressure. Okay. And at this fag end, where it is meeting the venules, capillary hydrostatic pressure is equal to 18 millimeter mercury. These three values are exceptionally important, and all along this point. So, there is, if I talk about the capillary hydrostatic pressure, there is another pressure which is coming into play, which is called colloidal osmotic pressure. Okay. So, colloidal osmotic pressure, which is out here, it remains constant all throughout, that is almost out here. If you look at it, the colloidal osmotic pressure is 25 millimeter of mercury. Okay. And if I had to now, so this is basically bulk colloidal osmotic pressure, BCOP. BCOP. Okay. So, now your net filtration pressure, if you have to calculate these values I haven't put, all these green values I haven't put. So, your net filtration pressure is equal to, let's put the NFP is equal to capillary hydrostatic pressure minus interstitial hydrostatic pressure minus bulk capillary osmotic pressure minus interstitial capillary osmotic pressure. So, now the values, what people know is this value is known to you, which I am circling, this value is known to you, you do not know these values, IHP, ICOP. These values are extremely difficult to calculate. So, this value, interstitial hydrostatic pressure, it varies from different part of the body. In the brain, it is approximately 5 millimeter mercury, and in other tissues it is around -5, it is a negative hydrostatic pressure. Okay. So, let us assume this as around 5, okay, 5 to -5, and whereas interstitial capillary osmotic pressure, where it is approximately around 5 millimeter mercury. So, if you ensure that these two terms, you are ignoring these two terms, and it boils down to capillary hydrostatic pressure minus capillary osmotic pressure. So, now if you do the calculation here, it is capillary hydrostatic pressure is 35 millimeter mercury and your capillary osmotic pressure is 25 millimeter mercury. This makes it 10 millimeter mercury. Whereas, if you do it here, this is 25, this is 25, 25 minus 25, it becomes 0. And if you do it here, this becomes capillary hydrostatic pressure is 18 minus 25, that makes it minus 7. So, if you look at these three values, that will give you a very simple idea is exactly what is happening. So, this is the zone where net filtration pressure favors NFP, favors that nutrients goes out of the vessel. So, in that process the oxygen is getting downloaded, because NFP or net filtration pressure is 10 millimeter, positive 10 millimeter mercury. Whereas, at this zone, if you concentrate at this zone out here, it's good, I put this line, you could demarcate all the three zones. There is hardly any exchange taking place, because it is here the pressure difference between inside and outside is 0. So, if you remember in the membrane physiology we talked about, it is the pressure difference which ensures the filtration is taking place. If there is no pressure difference, there won't be any filtration taking place. So, at this zone where it is 25 minus 25, you cannot expect any filtration to taking place. This value and this value ensures. So, the colloidal osmotic pressure and capillary hydrostatic pressure are equal. Whereas, if you move to this zone where it is in the close proximity of the venules, it is in this zone where colloidal hydrostatic pressure is smaller, it is approximately 18, whereas colloidal osmotic pressure is 25, and that gives you a total of minus 7 millimeter of mercury. This is the zone where which ensures that different stuff which are present, which has to be thrown out by the body, is being picked up by the capillaries in close proximity of the venules, and this is where carbon dioxide is being uploaded into the blood vessels, and which eventually reaches along the venules to the veins, to the vena cavae, to the heart, to the right atrium, then to the right ventricle, and then sent to the lungs, where it is being downloaded. And again we will come back, all these forces will come into play while we will be talking about the lungs, how the exchange is taking place in the lungs, how in the lungs the carbon dioxide is being downloaded and oxygen is uploaded from outside. That is the reverse process which takes place in the lungs. And again all these forces will come into play there. Okay, we will talk about it. So, this is an overall simplified summary of the exchange process in the capillaries for downloading oxygen and uploading carbon dioxide and ensuring any kind of other exchange processes. Okay. Here, if you look at if you come back to the diagram again, and if you look at the diagram, what is very important to ensure is that if, say for example, some XYZ toxin is coming into play, okay, say if this is the toxin, if this toxin reaches at some point and kind of change the permeability all along these places, then this is the zone where all these forces, all these capillary hydrostatic pressure, capillary osmotic pressure, all these are getting disturbed, and whenever there is a disturbance in all these things, the possibilities are there will be some fluid accumulation or some kind of a fluid accumulation at some point, and this fluid accumulation is what you call edema. So, now on, whenever you will see a edema or some form of accumulation of fluid, always refer to this diagram and try to request the doctor or ask yourself what is the capillary hydrostatic pressure, what is the colloidal osmotic pressure or capillary osmotic pressure, okay? What is the interstitial hydrostatic pressure, what is the interstitial osmotic pressure. As long as you follow this simple equation of understanding, where net filtration pressure is equal to capillary hydrostatic pressure minus interstitial hydrostatic pressure, or in other words, this is the net hydrostatic pressure. If this whole sum term I have to put it in terms of simple hydrostatic pressure, and this, one second, sorry for the interruption. And if this whole term has to be put together, then this is called the osmotic pressure. So, if you look at it, this very simple terms, hydrostatic pressure, osmotic pressure and all other details, these are very simple terms, and they ensure that your body is in perfect homeostasis. So, in other words, if I had to put it in terms of, so basically filtration is equal to hydrostatic pressure minus osmotic pressure. This is the broad term which describes this whole process. So, with this I will move on to the last tail piece of this, which is basically the venous pressure and venous pressure and venous pumps. So, again I will take you to a practical situation which will help you to appreciate this. So, you must have seen that at times in the schools where, or you are standing in an assembly, you are standing in a like kind of very attention position, some of the students faint down. Why that happens? Have you wondered? Is it some problem in the brain or something? These are very simple problems. So, what is happening? Think of it. You stand up and look at yourself. Your blood from the rear part of the body, from almost from the legs or something, has to come back to the heart. So, it is doing so, and especially along the vessels, of the veins, because for the arteries it is easy. Already there is a huge amount of pressure or force with which it is being pumped, and it goes all the way down and up of course, but there is a huge amount of pressure involved in it and it flows through. But in the veins, the only assistive mechanism is the valves, which does not allow the black flow. But the blood has to flow against the gravity at times. How that is being taken care? There are two distinct forces which takes care of it. One is called muscular compression. Muscular compression is all this muscle compression and all this taking place, that generates sufficient pressure for the veins to ensure that the blood comes back to the heart. But say for example, you are standing in an attention position and none of your muscles are really activated at that time, and you are in a standing position, there is a possibility that because of lack of muscular compression, the sufficient force which is needed by the veins to ensure that the blood flows fully to the heart is being reduced. So, that simultaneously leads to a reduction in the cardiac output, and that reduction in the cardiac output causes your fainting. So, next time whenever you see a friend of yours fainting standing in an assembly or some prayer meeting or something, ask them to just relax and kind of much more easy position, because it may, because of the lack of muscular compression, they may faint down, and especially in a bright sunny afternoon of anywhere in the tropics or subtropics, this is fairly prevalent. Okay. So, there is another pump which helps us to ensure that the blood flows through the venous veins, that is called the respiratory pump. That basically generated because of this. You see the whole chest is moving. That respiratory pump ensures that the venous blood comes back to the right atrium without much delay. So, these are some of the basic forces, some of the very basic mechanisms by which our whole complete network of the cardiovascular system works. I did not go into the anatomical details, but that you can go through. That is much of academic interest to understand what are the different names of the different arteries. But what is more important than that is to understand the mechanism by which it does. So, with this I will close on this part of this section of cardiovascular physiology, but while we will be doing the nervous physiology, and of course, I will scoop out certain time to talk about the hormonal control of the blood flow and the nervous control which ensures blood flow, and another situation which we will be talking about how those are helpful in ensuring that we do not suffer from some kind of edema or something, and how the anti-inflammatory drug works. As we will proceed through the course, probably once we will read the tail piece of the course, we will be able to understand all this by ourselves, as I will kind of regurgitate these things and kind of try to analyze all the situations. So, what is most important as we are proceeding through the course is that you people please try to realize our day-to-day life situation, what exactly is happening, because that is the best way to learn physiology. I can tell you a whole bunch of formulae and situations or diagrams or anything, but in your visual plane or in your mental plane, you have to have a visualization of the situation, what possibly is happening, and that is how the most of the bigger discoveries are being made. When people see something, try to rationalize in terms of simple physics, chemistry and mathematics, and then come back to biology and explore it and make some very fundamental discoveries. Okay, I will close in here. So, in the next section, we will be taking up partly the muscular system and the nervous system. Okay, thank you.